Fantastic. So I'm going to kick off by just reminding myself and everybody that we conducted a survey in September, partly of these meetings and partly of some of the tools that we had been talking about and putting some guides together for. And the PK tool was one of the things that we asked about in the survey, and quite a lot of people, this was question 5, said that they didn't have tools or resources to help make clinical or in vivo efficacy study dose predictions, or that they thought it wasn't applicable to them. So it was quite a small proportion of people that had the tools necessary, and also it seemed that the knowledge of the PK tool, which the foundation sponsored the creation of, was fairly patchy. Quite a large proportion of people were unaware of this, and perhaps that was partly because we actually talked about this, or more accurately Xenologic talked about this and launched it last year before we had started these global health surveys. So we thought it was timely to have a meeting to explain what the PK tool is about, and so that's really the purpose of this meeting. So I'm going to talk about where you can go for more information. This is only an overview. There is quite a lot more information, and of course information on how to download the tool. I'm going to talk a little bit about PK and the 2 methods of analysis that are possible in the PK tool. Apologies to those of you online that know a lot more about this than I do, but I think just trying to get everybody to the same point. And then I'm going to talk a little bit about allometry, which is one of the methods the PK tool uses, and then prediction from in vitro before we go to Gav's presentation. So, as I mentioned a little bit in the introduction, the tool was developed by Xenologic, which is now part of Certara. So I want to credit Neil and Cesar and Tomomi, who did a fantastic job putting this together. And also I have borrowed some of their slides. You can find all of their slides in the relatively extensive documentation which is there. So I've added a link, because I'll give you the slides in the follow-up email. There's a link to that introductory document which is a PDF with embedded links to lots of other information, including the installation instructions for PC and Mac, some theory slides, and also a presentation. There's some slides on hands-on training and also a presentation that they gave, and there's also links to test data which you can use, and the source code if you want to look at that. So in case that link doesn't work for you in the slides, that's the link for it there, and then the installation instructions are in a Word document here. So just to kick off with a simple slide on pharmacokinetics, and I suppose it's not really a full PK/PD analysis, but anyway, PK/PD light perhaps. So pharmacokinetics is obviously often characterized as what the body does to the drug, and you can see here a cartoon of a plasma concentration over time, this following an oral dose as a typical kind of profile where the absorption takes a certain amount of time, plasma reaches a maximum concentration and then decays away over time. Hopefully your plasma concentration is for a significant time above the minimum efficacious concentration, which is denoted there by the green line, and hopefully also you keep below the concentration, a plasma concentration that might give you some concerns about safety. So that's often you're trying to keep Cmax below that threshold. So the kinds of things that the PK tool is aimed at answering is what does that plasma drug concentration time course look like after each dose, and what plasma concentration do we need to achieve for how long, and also what, as we talked about, what concentration might possess a safety risk. So there are 2 fundamental approaches to trying to do this with the PK tool. The first is allometry, and actually this goes back to the 1930s, and essentially an observation that metabolic rate of a whole range of different species tends to, the log of the metabolic rate is proportional to the log of the mass to the 3/4 power, and the clearance very closely linked to metabolic rate. And so in the PK tool it actually uses unbound clearance, we'll talk about binding in a minute, to the 3/4 power, and that's a standard allometric technique, and perhaps one of the simplest ways of making a dose prediction. The other method that you can use in the PK tool, this really is a prediction now where you're using an in vitro clearance estimate. So you still need to have some parameters from PK ideally, but the clearance estimate is now provided by some experimental in vitro data, usually liver microsomes or hepatocytes. And as I've characterized here, the microsomes are usually considered only to give you some information about oxidative phase 1 metabolism, much of that anyway, and hepatocytes can include some phase 2 clearance mechanisms such as glucuronidation as well. One thing that we need to get into a little bit is plasma protein binding, because the PK tool is essentially built on the standard methodology, which is to use unbound clearance and volume for both allometric scaling and dose prediction. So basically, apologies for dumbing this down a little bit, but the free drug hypothesis essentially is that only the free drug moves across cell membranes and binds targets and is available for clearance enzymes. So it's really the concentration of the unbound drug that is key, and obviously the fraction unbound, which is what's driving the activity, can vary from nothing to 1. So that's not quite right, is it? Fraction unbound of 1 would, yeah, I'll change that slide. You can see what I mean. Fraction unbound is what drives things. So if it's all bound to protein, as characterized on the right-hand side, then there's no free drug. And in terms of binding, it can be saturable, so that depends on the binding of the protein, depends which protein we're talking about, and there can be marked differences between species. So measuring binding in different species, the species you're interested in, is important. In terms of an orientation to the PK tool itself, this is what the screen looks like. So it does have a glossary within it, so you can see these tabs on the top left here. One of them is glossary, and you can click through for terms in vitro, in vivo, and some of the calculations. So there is some online support to understand some of the acronyms and what some of those terms mean if you look over those. And also there is a hover function. So if you hover over things like the text for, apologies, this is so small on the screen, but this is hepatic clearance, hepatic cell rather, it'll tell you what that box is expecting. So it gives you a little more guidance about what numbers are expected in these boxes. Essentially, this top left panel is where you input the values from experiment. So from in vivo and in vitro ADME and PK in the top left here, and you can see that there are tabs for in vivo and in vitro for multiple species, and in fact there's another tab here, so you can define the parameters for any species you like, in fact. So the experimental values that you put in here generate graphical summaries and key numerical values on this top right-hand side. which we'll have a look at in a bit more detail. And then you combine these key parameters with options that you choose down here. So these are options like the dose frequency, molecular weight of the compound, C efficacious, C safety, that kind of thing. And they produce the dose prediction and a graphical output of the plasma concentration time course down here. So we have a look at a first example. This is using allometry. So the tool does what's called single species allometry. So basically you're projecting from a single species at a time. You can use whichever species you like, but it doesn't try and do an aggregate across species. It predicts human dose from a single species at a time. So in this tool, the exponent is fixed to be 0.75, as we talked about, for clearance, and 1 for volume, and also for and Ka. Actually, Ka is fixed. So the intercept is then calculated from the input species weight, so which you can select which of the species drives the allometric scaling, and it's unbound clearance that's scaled in this in the PK tool. So there's some further information about this approach in this reference here, and that's you'll see other references pop up through the slide set. So I mentioned that there is some there's some idealized test data you can pull in, and it's in the form of a CSV file, so you can have a look at it and see the kinds of things that are brought in. You can write these files out as well as reading them in. So import parameters is the parameter file that we're talking about here. So once you install the tool, you can also download testinputs.csv. That's available from the documentation that I referred to at the start. So you can read these files in. If you change values or put your own values into the PK tool, you can then also export the parameters as a CSV file. While we're here, exporting the simulation data as a CSV, that's quite a large file. What that is doing is exporting the graphical output at the bottom rather than the parameters. So thinking about allometry, the key inputs here are the in vivo endpoint types. The only other bit of information we require from the in vitro is actually the free fraction in plasma. So when you read in the data, the tabs are populated. Here I'm just showing the mouse in vivo data tab. So various parameters, the volume, the clearance, and the absorption rate constant. This is the weight of the animal, free fraction, and the bioavailability. And then on the top right-hand side, when calculate is pressed, then you get some calculations carried out for each of the species where you've got experimental data. So here you've got the mouse in vivo data, and the tabs here are calculating the unbound clearance values. So in this case, the unbound clearance is just the total clearance divided by the free fraction. So that's what you see here. On the graph, and I'll show you this in a minute, the graph, because it's allometric scaling, that's clearance and volume for the body. So you adjust the unbound clearance here for body weight. Okay. So just to show you that, so these three graphs at the top, one for Ka, one for volume, one for clearance. Volume and clearance here are unbound, and they are per body, okay, because it's allometric scaling that this graphical output is producing. So you can see the weight of the species along the bottom, from mouse all the way to human, and the blue stars here are the in vivo experimental values that you've got adjusted for binding. Okay, so these are the unbound clearance values for the body. These are the unbound volume, and this is the Ka. And so the allometric prediction is the line. This is scaled by mass to the scale by mass. This is scale by mass, log mass to the power of 3/4, and this is actually, you can't see it, but it's a straight line. So the human prediction is assumed to lie on the allometric line at the body weight of a human, which you can adjust, but here is assumed to be 70 kilos. Okay, so you can, using this tab, you can change which species is being used to derive the allometry. We'll see that in a minute, but by default it's set to minimum weight, and in this set of data the minimum weight is mouse. So that's what's driving the allometry, and that's why the allometric line goes through the mouse value. In the table at the bottom here, the values on the graph, as I said, are per body, and they are they're unbound. Values in the table here, in the theoretic allometry column, these are per kilogram, and they are total. Okay, so to convert from the table to the graph, you need to convert for weight and for binding. So you can see that the clearance value here of 0.5 mils per min per kilogram. To convert that to find its position on the graph here, we convert for weight and convert for the free fraction. So we end up with 1500 mils per min, and that's where the unbound clearance for human. Okay. So as I mentioned, you can adjust the species that's used to drive the allometry. So here, if you choose rat, now the lines all go through the rat in vivo values, and obviously that changes the positions of the lines. And you also get, so for other species, so for example dog here, you get the user data column. This is basically just a recapitulation of the user data that was put in on the top left, but now unbound. And you also get a theoretical allometry column, so you can compare where the allometry through the rat in vivo data, how that compares with the dog. It's making an allometric prediction for dog, basically, based on rat. So you can see how those two things compare. So just in terms of where we're up to so far, we haven't made a dose prediction yet. We've been looking at the input values from the top left and the graphical summary and the table of information, or the tables of information on the top right here. And then we need to combine that with options on the bottom left to do with the drug and dosing frequency, and then we'll have a look at the graphical output. So far we've got predicted values for human of Ka and volume and clearance from allometry, and then we need to know something about the bioavailability. This is quite a hard thing to predict. Might easiest be obtainable by looking at the bioavailability for other species. The free fraction in plasma, hopefully you've measured that in vitro, and of course the weight that you're predicting for. And then finally, the other parameters that we need to add in are the efficacious concentration. So there are kind of, that's often the minimum concentration of drug at the trough required to give the desired effect. So the assumption here is that in order to drive the biology, you need to keep the concentration of your drug above this efficacious minimum concentration. Alternatively, you could modify the CF to give you a target AUC. So within the tool, you can't set AUC as a target, but you can, if you want to, play with the CF value to give you a target AUC and then a predicted dose that way. Okay. C safety, as I mentioned, that's basically just going to give you a red line at that value. So it's really, it doesn't affect the calculation at all, but it serves you as a warning if your plasma concentration goes above the safety line. So you might get that from things like HERG or cytotox, and oftentimes you want to keep Cmax below that. The other things you need to input: molecular weight, the simulation time, dose interval, and the route of administration. So then the tool calculates predicted human dose, and the calculation is presented on a mg per kg basis. So obviously if you want to know total dose, you've got to adjust for the body weight. And of course, so this value here is based on the input CSV file that I showed you, and is assuming that you used mouse for the allometric scaling. As I mentioned, this is a sort of idealized data set, but if you use rat or dog, you'll see how the predicted dose changes. You will also see down the bottom here, there's a confidence in prediction given, and that's generated by an algorithm which is explained in the reference material. But essentially, to have a high confidence, you need at least 2 species PK which are in agreement with each other, and in agreement with in vitro, or 3 in vivo species. So high confidence is quite hard to achieve without a lot of data, but that's because PK prediction is quite tough. So we're trying to be realistic here. To get a high confidence prediction of PK is not easy and requires quite a lot of data that all aligns with each other. So that was predictions using allometric scaling. We're going to look at now predictions using from in vitro data. So this is using in vitro data to provide predictions of clearance. So we discussed that liver microsomes generally give you information about phase 1, oxidation, clearance mechanisms. Hepatocytes also have some phase 2. Obviously there are other clearance mechanisms, so clearance through the urine, other ways in which drugs can be metabolized. So in terms of taking clearance predictions from in vitro systems, it's quite often true, and the OBAC paper, which I reference in a minute, makes the point that oftentimes these clearance predictions are below the true clearance, because the true clearance, the true in vivo clearance, is often a sum of several clearance mechanisms, of which only part is likely to be microsomal or hepatocyte clearance. So in the PK tool, you choose whether you're going to do your dose prediction from allometry or using the clearance mechanisms from in vitro using this radio button here. And if you choose in vitro, then you get to choose between hepatocytes and microsomes. So as I mentioned, it's only the clearance estimate that is derived from these additional hepatocytes or microsomes. The other parameters on this human X parameter tab are coming from the well, they're taken from that tab. Where you get them from, you can put your own numbers in, but they're ideally derived from a PK experiment. So we've touched on the idea that the assay systems are liver microsomes or hepatocytes, and what we calculate is the intrinsic clearance and also the blood clearance. So this is the one that's most closely aligned with the total clearance used in the calculation of the that the PK tool uses. So from looking at going from microsomal data to the clearance blood that the PK tool uses, this Scott Obach paper is used. The calculations in there are used. So we calculate intrinsic clearance. Basically, this is taking the in vitro half-life in the microsomes and adjusting for the microsomes that are present in the assay, and also making some assumptions about the amount of microsomes per gram of liver, and also the liver weight per body weight. And then the blood clearance makes adjustments for the binding in the blood and also binding in the microsome prep. And this is one of several methods that are referenced in Obach's paper, but the one that is one of the ones that's preferred in that paper. When you're putting these numbers in, you use the human in vitro tab if you're putting in the human in vitro data. So these are some of the system parameters that go into doing the calculation. These present here, you can put in either hepatocytes or microsomes. So here are the system parameters that are needed for both calculations. I'll come on to hepatocytes in a minute. So these are populated for you for the different species, or you can change them to your own preferred values if you want to. So if you have half-life from your microsome, in vitro microsome experiment, then it will calculate the Clint for the microsomes for you, and also clearance blood. But if you have a Clint value, you can put it into this box, and you don't have to bother then with the some of the system parameters to calculate the Clint. So you can either go from half-life or from Clint. And then in the human in vitro tab, you'll find the Clint values if you've put in microsomal half-life in minutes here, or just clearance blood if you've started with Clint. Alternatively, you could start with in vitro hepatocytes, and this is a paper that was used as the reference for this. So in doing the calculations here, we first calculate clearance per million hepatocytes. So the first calculation is to adjust the half-life to give you a clearance per million hepatocytes, and then in a similar way to previously, we're going to calculate Clint as milliliters per min per kg based on the number of cells per liver, and also the liver weight per kilogram of body weight, and then converting to blood clearance similarly as for the microsomes. So again, the parameters that you need to enter, you can either enter the hepatocyte half-life and calculate the Clint using the tool, if you know things like the incubation volume, or you can put in the Clint directly, and the calculations here are displayed here. Just to note that this is a typo. This top one is milliliters per min per million cells, and the rest are mil per min per kg. Okay, so actually using the tool, an example of using the tool for in vitro clearance estimates. So if you look at the data set that's read in, there are hepatocyte Clint values for several species, including human. So that'll be present here. And in this case, it's not derived from half-life. It's derived from the calculations based on the Clint values already been calculated. So, as I mentioned, the clearance estimate for the dose calculation is based on the Clint value here from the clearance blood value from hepatocytes, human hepatocytes, and the other values are based on those from the present on this tab, based on allometry still, so Ka and volume coming from here. So if you choose to do the calculation from in vitro and from hepatocytes, values for Ka and volume are still coming from this tab and F, etc. Okay, and in this case, with this idealized data set, you get a value of 17 mg per kg rather than, I think it was 5 mg per kg based on the allometry. But you know, this is an idealized data set. Okay, when you are looking at one thing that the tool does for you, which is nice, is on this allometric graph here, it adjusts the all of the clearance estimates onto the same allometric scale here, so you can see how the in vitro estimates compare with the in vivo estimates. Obviously, being an idealized data set, it looks as though all these estimates are pretty much in line with each other. Not always the case. And quite often you might expect to find the in vitro estimates are significantly below the allometric scaling line, and that might be an indication that non-hepatic clearance is a significant component of the route of clearance, so that you'd probably need to investigate the other clearance mechanisms. One final example I want to give is that obviously the primary driver for putting the tool together was to make human dose predictions, but in projects it's obviously one of the before you get to that point, you may well be selecting compounds for efficacy studies in obviously non-human species, and at that point you often have a lot less information and sometimes even no in vivo PK, or certainly not necessarily in vivo PK on the specific compound you want to predict. Obviously this is going to mean that the predictions are going to be pretty uncertain, but you know, what can you do to provide some estimates? So for the parameters that you're going to need, these are the 6 parameters that are going into the dose prediction. You need an estimate of plasma protein binding, or the free fraction in plasma, and as we'll see in a minute, there is often a fairly good correlation with lipophilicity, though you need to check your own series, of course. And volume also can sometimes give a reasonable correlation with lipophilicity, and I'll just show you that for a series I've been working on, quite lipophilic series, it has to be said. On the left-hand side, you can see the free fraction, log of free fraction plotted against log P, and that really does give quite a nice correlation. And similarly, the log of the unbound volume gives a reasonable correlation with log P2. It's fair to say that most of this straight line is really driven by the binding, and the volume is sort of 3 plus or minus threefold. Okay, but I guess what it indicates is we could, you know, in extremis, make an estimate for the free fraction based on lipophilicity for this series. Then obviously clearance, you're likely to make predictions from that from in vitro measures of microsomes or hepatocytes, as we've just been talking about. You do ideally need a blood plasma ratio as well. You can assume that sometimes it might be reasonable to assume that that's 1. That's an option. And bioavailability, as we discussed briefly, that's pretty hard to predict, partly because it's a combination of factors, so derives from fraction absorbed as well as the fraction escaping intestinal metabolism and the fraction escaping hepatic clearance. So it's a composite figure, so really it is quite hard to predict. Certainly CACO2 and MDCK and those kinds of measures of absorption can give you an indication when the fraction absorbed is going to be low, so you can certainly get some ideas that bioavailability may be low if you've got very poor flux. But just because you've got good flux doesn't mean you're going to have necessarily good bioavailability. So ideally you want to have some idea of what your bioavailability is going to be like, either from that compound or perhaps from analogues. If you want to look at the rest of the parameters and how good it could be if bioavailability was good, if you set it to 1, I guess you can get an estimate of what's the best it might be. But certainly your bioavailability may very well be very far from 1. Ka, this is the absorption rate constant. You could take a look at analogues if you've had those measured in PK. You could assume it's 1. It varies between sort of somewhat less than 1 and up to about 10 or so. The impact here, what does Ka do? The impact is mostly on Cmax and Tmax. So for high values of Ka, Cmax might be a bit higher, so you get a higher peak, and the Tmax might be a bit earlier, but it tends to affect the AUC a little bit less, and the trough concentration a little bit less. So it depends what your objective is. If you assume it's a certain value, you're probably not going to get a very realistic estimate of what Cmax or Tmax might look like, but for dose prediction it might not be too far off. And then the PK tool assumes that you're dose predicting for humans, so you can change the weight, but you've got to remember to change the weight because your efficacy species is probably a good deal lighter than 70 kilograms. So this is just showing that you can, in the tab here, this is the human extrapolation parameter tab. You can, by checking this box, you can put in your own values here and derive them, as we've just been discussing. And then you can see here, this obviously isn't a human, make a dose prediction for your efficacy species. Obviously the tool won't tell you that it's made a prediction for something other than human, but you can get the tool to do that for you. Finally, I just wanted to have a couple of slides on manipulating the plot in the output. So some of the things that you can do, you can change the Y scale in this PK plot to a log scale if you want to. One very useful feature is that by default it's single run under here, but you can carry out multiple runs so that you overlay a new trace on the old. So for example, if you fix the dose, which you can do by checking the dose box, you can overlay 2 different doses and look at that, or that's a very useful feature. You can toggle between free drug and total. Oops. You can, you may have been finding some of the font hard to read. You can change the font size and you can also manipulate the X and Y scales here. So at the moment it's 7-day simulation, and the plot is for all 7 days, but you can change the X-axis to look at a portion of that, or you can focus in on a part of the Y scale as well. So some of the kinds of what-if questions, there's lots you can come up with, I'm sure. We've discussed the possibility of looking at the impact of varying the dose. So if you, with the default parameters on the input, you'll find that the dose is predicted to be about 5 mg per kg. You can click this, check this box, and then put your own dose in. And by using the multiple runs, you can overlay what a 10 mg dose would look like. You could also look at varying the dose interval. So by default, or at least in that input table, it's 24-hour dose interval. You can look at a 12-hour dose interval. I've only shown you the first couple of days, and basically this predicted dose, the objective is to get to CF by the end of the 7-day period, and you can see, because the half-life is quite long, you're seeing build up over the first couple of days. So you can see the impact of that here. Last slide, I think, on caveats. So it's a relatively simple tool for PK prediction, and there are more complicated ways of doing it. So this is it uses a one-compartment model. So what you would see if that were literally true, if it was if the plasma concentration was well explained by a one-compartment model, would be a linear line here on the log of plasma concentration. You can see that this is not really a one-compartment model, or not well modeled by a one compartment. So 2 or more compartments might be better in some circumstances. Again, the AUC is more or less preserved in a one-compartment model, because often the half-life is based on the elimination phase here rather than the distribution phase, but the terminal half-life might be different. So it depends a little bit. It's worth looking at your PK profile as to how well a one-compartment model is going to work for you. As I've tried to indicate, obviously extrapolating a PK to man can be non-trivial. Plenty of other things can trip you up. The absorption prediction is uncertain, PK variability between species, active processes, so a whole host of things. Hepatic extraction may not be the major clearance mechanism. That's certainly one of the larger things. So there's plenty of reading, and I guess get out of jail free card. Always advisable to consult experts. Even if you are using the tool, I think it's very reassuring to speak to experts and get some input from them that what you're doing and how you're using the tool is appropriate. Okay, so I'm going to stop there and just check very briefly if anyone has any questions before handing over to Gav. Okay, that's fine. Gav, let's see if I can make you presenter.
Okay.
There we go.
Okay, thanks, Mark. Okay, so hopefully, Mark, can you see my slides?
Yep.
All right, great. So I'll assume everyone else can as well. So Mark's given you a really great introduction to how the tool works and the kind of inputs that you need to use it. What I want to do now is just take you through 2 or 3 examples of how we've used the tool in a number of projects that we've been running in collaboration with D&DI. All these projects are sort of anti-parasitic projects under one of D&DI's lead optimization consortia, all focused on new treatments for visceral leishmaniasis, trypanosomal parasite. You've already heard how critical it is in any drug discovery project to understand the relationship between the in vivo exposure that drives efficacy in your models, what your preclinical PK looks like, and therefore using that information to try and predict as best you can what a likely human PK profile looks like and what an efficacious dose might look like. What I want to do is take you through a couple of examples where we've used rat allometry to assess PK and dose prediction, and then one further example where we're going to go into where we've got data on 2 species and how that brings in, well, that could bring in uncertainty, or it could give you increased confidence as to what your prediction in human might look like. So looking at example 1, so all these compounds have been done using rat allometry, so we've generated a lot of in vitro and in vivo data to give us confidence that these compounds are worth taking forward to rat IV and PO PK studies before we start looking at predictions. So what I want to do is just walk you through a little bit of the in vitro data that then drove our decisions to take these compounds forward. So this example's what we'd call an advanced lead compound from a new series, and the key question we wanted to answer for this compound as an exemplar for the template was, can this series achieve what we think will be an acceptably low human projected dose? The data that got us to this particular point was very potent in vitro activity against the 2 species of Leishmania parasites that we're interested in, so 150 to 300 nanomolar activity, good stability in vitro in terms of human liver microsomes and rat liver microsome stability, moderate stability in physiologically relevant media, so fad and fed simulated gastric fluid at various pHs, and we've also checked in vitro permeability to make sure that we think the compounds are going to be reasonably permeable. All of that data then led us to do some efficacy studies, in this case in the hamster, looking at reductions in levels of L-infantum and L-donovani over a 5-day dosing period, and we found that the optimal dose level for this compound was 25 mg per kg, and we can see what we're looking for here to meet our target product profile essentially is anything above 95% across all of the different organs where we're looking for parasite reduction, and this compound certainly met that particular criteria. So the key question now is, well, combining all this data that we've got in the hamster in vivo, when we run rat PK studies, what does our human dose prediction look like? So the key pieces of data are, well, what's our efficacious drug concentration? Mark in his talk, he talked about how you can use an AUC as your efficacious exposure rather than a C-min, and that's certainly what we've been doing for this project because we think that's the most relevant number. So we've measured a free efficacious AUC over a 24-hour period to get this number. We've generated IV and oral rat PK data, so the compound's got pretty low clearance, about 10% of liver blood flow, moderate volume because it's essentially a neutral compound, and moderate bioavailability, and some of that might be linked through to its not spectacular solubility. So we've gone through and used the tool just as Mark described, inputting the rat PK data, inputting the plasma protein binding data for rat and human, and I'll show that on the next slide. And what we get out of that prediction is a low predicted clearance, which we're certainly not surprised about based on the relatively low clearance in rat itself. A relatively short half-life, so we'll certainly be looking at a BID dose for this particular compound, but the initial predicted human dose for this compound is quite low. It's around 5 mg per kg, and that's well inside the kind of values that we'd be looking for taking any potential development candidate forward from this template. So at this point, we've got reasonably good confidence that not only this compound, but as an example for the series, that it's worth progressing with the series. We can deliver compounds that are both potent enough in vitro and potent enough in vivo, and can have sufficient PK to deliver a compound with the right kind of profile. So just flipping back to what the tool looks like again for a second, because we're looking at an efficacious AUC rather than a C-min, this is the box. This CF box is the one that we just dial up and down to then match what our efficacious AUC is here. Once we're happy that figure's right, then that gives us our approximate human dose prediction in mg per kg. So that's one example where we've got really quite a low free efficacious AUC The compound's very potent in vitro and has got a reasonable PK profile. It's low clearance, but only moderate bioavailability. What we then found for compound 2, which is from the same broad template, was an in vitro profile that's relatively similar, but some significant differences in its profile as well. And we've kind of gone through the same process to then use the tool to understand what's the downstream impact on what the human PK and dose prediction might look like. So the in vitro potency for this compound is very similar. The in vitro metabolic stability is very similar, but we've seen a very significant difference in physiologically relevant solubility, so around up in the hundreds of microgram per mil now. But we are seeing a slight difference in MDCK permeability as well, so we're starting to bring in a little bit of PGP efflux, and we're only going to start teasing that out when we start running in vivo studies. And in terms of efficacy, you'll note from the previous compound that we had an efficacious AUC of around 550, and this time the value's up closer to 12,000. So the compound in vitro looks quite similar, but clearly something slightly different going on in vivo. Compound needs significantly higher free drug concentrations in vivo to work. What impact is that going to have on what we think our human dose prediction might be? Now, in terms of rat PK, this compound's actually got slightly lower total clearance, but it's much more bioavailable. So the last compound was only about 35–40% bioavailable. We're up at 80% now. So we're reasonably confident that that's driven by solubility, and possibly this degree of PGP efflux isn't having a significant impact. And working through those numbers again using the tool, again this compound's predicted to have very low clearance in human, and combining the low clearance prediction actually gives us a really very sensible looking dose prediction. So even though for this compound you need significantly higher free levels to drive efficacy, because of the dramatic increase in bioavailability, those two things kind of cancel each other out, and we can still achieve very acceptable human PK predictions. So I guess for us this was important, so for when we see compounds that maybe at the face of it don't work as well in vivo as we'd like, just by sticking with them and working through the numbers, and maybe running an extra couple of studies to understand how good they are, then you can make, I think, slightly more informed decisions about whether you take that compounds that light forward, and certainly emphasizes to us that the parallel optimization of potency and PK has been really important in taking this series forward. So I guess the caveat to both of those examples is that this is based purely off rat allometry, and as you've heard from Mark, that would kind of by default, using the tool, give you a low confidence in prediction. So ideally it would be great to be able to generate a little bit more data to either increase your confidence or highlight a potential uncertainty going forward towards clinical development. And that's what we've got in example 3. So again, this is an advanced lead compound from an interesting series where we've generated rat and dog PK data, and the key question here is, do those two predictions align? Are they different? How do we feel about that data? In vitro, this compound looks well behaved, it's potent, it's got pretty good stability across the board, very similar in human and rat and dog liver microsomes. Okay solubility, no significant efflux, so we're confident that the compound should behave reasonably well in vivo. It's efficacious, and in this particular case, this is a key number, so an efficacious AUC of around 1700 hours nanogram per mil. And what we find though, when we take this compound in vivo, we see quite different effects in rat and dog. So clearance in rat is relatively low compared to liver blood flow. It's around 10 to 15%. Quite low bioavailability, which again might be driven by the moderate solubility. Dog PK is quite different though. So this compound looks pretty much liver blood flow clearance in the dog, so rapidly cleared. We're still seeing a little bit of bioavailability. So working through both of those scenarios using the tool, so rat allometry and dog allometry, gives us two very different situations. So if we're being optimistic and we like the prediction from rat, then the compound should be low clearance in human, with a clearance of around 2 mils per kg. Alternatively, if we're being pessimistic, then we might think, well, the dog's predicting essentially liver blood flow clearance in human, and why would we take that compound forward? And by working through both of those numbers through to a dose prediction, then you end up again with a tenfold split. So low dose based on the rat, high dose based on the dog. And then it becomes a case of, well, what do you do in that situation? We've got a high level of uncertainty around the human clearance prediction and the dose prediction, but we didn't stop that compound on the basis of that data, and we certainly wouldn't advocate someone just looking at that data in isolation and saying, oh well, there's too much uncertainty, too much risk, you shouldn't go forward. But it has kind of prompted us to think about what do you do, and options certainly would include further in vitro studies to look at routes of metabolism. For example, does the route in vitro in rat look more like human, and does dog produce a specific metabolite that is unlikely to be a pathway in human, and therefore you could possibly believe the rat data in more detail, or vice versa? Do you think in vitro that dog is more like human, and can you build a package of data that says that you should stop that compound? What I would say is that just by running additional in vivo studies in more species, for example, mouse or primate, is unlikely to increase your confidence, because we're still going to end up with a large range of clearance predictions. So my experience of having done this for a couple of projects within the big pharma industry is that it doesn't really help your decision making. It just slows you down by 3 to 6 months as you generate the data. And ultimately, if it's a compound that meets enough of your criteria, despite that uncertainty, really the only way to fully understand your human PK is to run a human PK study and know for sure whether the compound's then worth taking forward into further clinical studies. So, that's been a relatively whistle-stop tour through some of the examples and some of the ways we've used it. We very much focused on the allometry piece. We haven't done that much work in terms of the in vitro scaling. Certainly, I'd be really interested to hear any examples where people have used that successfully from anyone else out there. But from using it for rat PK and dog PK and other species for other projects, we've certainly found it to be really useful, because it's helped us understand the translation of both our efficacy data and our preclinical PK data into potential human PK dose predictions. Do they align across species? Do they give us cause for concern in terms of confidence and uncertainty? And early on in projects, when possibly you get the first signs of efficacy in a template, and you take a compound forward into a rat PK study, using those pieces of information to understand, well, how does this benchmark compare to where I need to be for the overall project, can then really help feedback into improving compounds in the future. Is it potency I need to fix? Is it pharmacokinetics, or is it a little bit of both? So we've certainly found it a really valuable addition to the way that we've been running these projects with DNDi. And that is about it. I'll be very happy to take any questions on that. I appreciate it's been a bit of a whistle-stop tour, so happy to take questions now or via email after the session.
Fantastic. Thanks very much indeed, Gav. I can see Cal has asked a question about MDCK and CACO data, which I've tried to answer by text. Has anybody got any other questions? Okay, we are bang on the hour. I apologize for my session running a little bit. You will all get email with links to the presentation in it, and feel free to follow up with emails to Gav or I. I'd just like to thank Gav very much for volunteering to give a presentation, and also to thank the guys at Xenologic for putting the tool together, and also for giving me some feedback on the slides. I can see Joshua had a quick question, but looks like it's gone. Okay, so please feel free to follow up by email. Thank you very much, everybody, and have a great holiday, and hope to see you again in the new year. Thanks a lot.
All right, thanks, Mark.
Cheers, bye.